fall starts, no delays due to high water. I think we're going to have a good summer. So we're looking forward to a full program this year. Chelsea, if you're counting noses, let me know when we've got a full complement.
You can go ahead.
Good afternoon, Alan. And good afternoon, water quality volunteers. We are so glad to have you. So Chelsea, can you start the screen share, please? So this year's program is going to run on 4 dates, May 20th to 23rd, June 24th to 27th, July 22nd to 25th, and August 19th to 22nd. Today, your program is going to be brought to you by Jan Getson, who's going to talk about the program itself and some tweaks that we've made from last year. I'm going to give an introduction. Chelsea Callard. Chelsea is back with us after having a leave and is going to be running the program from the office. There's her phone number if you need anything from the office. We have a new general manager, Erla McCaig. We have, as of last year, a new consultant, Chris Hadley. Chris is with Hutchison Environmental Sciences Limited. And our testing lab is the same that we've had for the last couple of years. Next slide, please. So here's our agenda for the afternoon. I'm just going to give a brief introduction. Jan's going to talk about minor changes to the program for this year. We're going to review the training video that was made last year. Then we're going to have a Q&A session. And to operate the Q&A session, please have a look at your Zoom toolbar, either the top or the bottom of your screen. You're going to see a button that says Q&A. If you want to put in a question, type it in there and we will answer those when we get to the Q&A portion of the program. There's not going to be any live verbal input in terms of you asking the questions. So we're going to read the questions and then either Jan and I will answer them. And if there's further follow-up, please type it in. Hopefully we'll get through all the questions and then we'll do a wrap-up. Next slide, please. So this year will be our first year return to normal. You may remember we didn't have any testing 2 years ago. And last year we didn't get off to a start until 2 or 3 weeks into the season due to COVID protocols. A week or so ago, the MLA put on a webinar to present last year's results. That was on the 10th of May. And if you're interested, that should be on the MLA's website if you want to replay it. Generally, the results were good. There were about a dozen yellow sites, which were about half due to algae and half due to bacteria. And our consultant pointed out that rainfall may have influenced some of the results. So we're going to talk a little bit about that when we get to the tweaking part of the program. We also had our first red site where we had a long-term trend to have increasing phosphorus. I got to tell you that that is the only place that that happened in the entire sampling program. And so we're going to undertake a little bit of extra work there to try and figure out why that happened. Next, please. We're fortunate this year that the water levels have generally been well behaved. And right now we're down just a few inches above summer level. We did not get high levels in the spring. And so everybody should be able to have their boats in. And now I'm going to turn the program over to Jan, who's going to talk about some program tweaks. Next slide.
And thanks to all of you for volunteering your time to this initiative. Before we watch the video, I want to highlight several parts of the protocol. Ideally, we want to sample when the water is calm and when there's no precipitation. If we have had more than 10 millimeters of rain, we prefer to wait for 24 hours. Unfortunately, this is not always possible, but we do try and check the weather forecasts in advance and pick the optimal window to do our sampling. Chelsea can put up this week's forecast on the screen so you can see that we do have a challenge for this coming weekend. The other thing that is important to remember is that we are doing a longitudinal study. And so it's very, very important that we are consistent in the locations that we go to make our data reliable. And we have geographic coordinates on the maps in our binders with our pictures to make sure that we are always in the right place. The big tweak that we are making this year as a suggestion is the use of the notes section on the data sheet. Our notes are sent to our consultant and they're relevant for interpreting the results. This is where weather conditions should be noted in the past. Please note that there has been intermittent rain over the past number of days. Note if there was heavy rainfall that caused erosion. Note if the wave action isn't calm, but it's not rough. And if you can estimate the height of the waves, that would be great. If you've observed a flock of geese or deer near your sampling site or construction that's new, this information can be very relevant, particularly if the result is an outlier. And finally, when we sample water at our deep water sites, we want to collect water throughout the water column. Therefore, we need to finish filling the collection bottle when it reaches Secchi depth. To do this, we first measure the time it takes to fill the bottle at the site. And Chelsea, we have a slide to show us how we do this. We time that, and then when we are collecting the water for our actual samples, we regulate the speed that we lower our Secchi dish to match this time with the collection bottle attached. Also a reminder that for our first collection of the season, we take a duplicate sample at our deep water site. We do use the same grab of water. Then we remember that although we rinse the collection bottles and the funnels, we do not rinse the sample bottles as they contain chemicals. This means we have to be very careful that we just fill those sample bottles up to the shoulders of the bottles because we can't empty them. Now let's watch the video, which will be followed by the Q&A, and remember to submit your questions in the Q&A box. Thank you. Good morning. I'm Jan Getson, one of the water samplers for the MLA, and along with my cohorts, Chris Craig and Eleanor Lewis, we're going to be taking you on a step-by-step guide of what you'll do when you get out in the boat to water sample. First of all, let's start with the kit. Each team will be given one of the kits, and each water sampler will have their own copy of the manual. I've taken my manual apart just to show you what's in it. First is the calendar of dates that we sample. Then we have the quick reference guide, which is the step-by-step reference for what you'll be doing, and that is what we're following in this presentation. For a better understanding, complete understanding of our monitoring program, you also have a detailed volunteer manual. Both of these should be read before you go out on the boat. In addition, each kit will have a map that is specific to their testing area, and it shows all of the testing sites. In addition, there will be a description of each one of the sites.
Looks like we're having a few technical difficulties. So we'll restart the video once we've ironed out what's causing it to skip.
Chris, there's a question in the question box as to whether or not you could restate the dates. So maybe while we're waiting, you could do that.
Okay, the water sampling dates. The first weekend is this coming weekend, May 20th to 23rd. The second weekend is June 24th to 27th. The third weekend is July 22nd to 25th. And the final weekend is August 19th to 22nd.
And all these dates are the first page in your binder. Good morning. I'm Jan Getson, one of the water samplers for the MLA, and along with my cohorts, Chris Craig and Eleanor Lewis, we're going to be taking you on a step-by-step guide of what you'll do when you get out in the boat to water sample. First of all, let's start with the kit. Each team will be given one of the kits, and each water sampler will have their own copy of the manual. I've taken my manual apart just to show you what's in it. First is the calendar of dates that we sample. Then we have the quick reference guide, which is the step-by-step reference for what you'll be doing, and that is what we're following in this presentation. For a better understanding, complete understanding of our monitoring program, you also have a detailed volunteer manual. Both of these should be read before you go out on the boat. In addition, each kit will have a map that is specific to their testing area, and it shows all of the testing sites. In addition, there will be a description of each one of the sites, which will has both photographic references as well as GPS. Then you have your data sheet that you will record and turn into your team leader for each testing period. Being familiar with this material, I'm getting myself ready to go out to test. I've started by filling in my data sheet with date, train sampler name, rainfall in the past hour, all of the information that I can do at home so that I'm not trying to write in the boat. Then I've assembled my equipment. This is the Secchi dish that we will be demonstrating later, which we use to measure clarity. You have a thermometer, a Sharpie. This is our sample bottle or our collection bottle. Filter and funnel. And the sample bottles. The important thing to notice about the sample bottles is each one is labeled, pre-labeled with the site, but you will need to write the date and on the back of each bottle each time you go out. For our first period, our first testing period, you will need 2 bottles per site, because for our first one we do duplicate samples. The other thing that I have here is a cooler and ice, a bag with ice in it, because we do need to keep our samples cool until they get to our team leader. Before we go out, of course, we will coordinate with the team leader to make sure that everything is going to be assembled. That's particularly crucial for bacteria sampling. We've arrived at our deep water site. We're just checking the coordinates with the coordinates on our chart, and we're fine, ready to go. Step 1, we'll put the thermometer in. Next step is to check for the clarity, and we do that using the Secchi dish, taking it down until we can't tell the distinguish between the white and the black. I'm standing on the shady side of the boat for both the thermometer and for the Secchi dish, and make sure that the sunglasses were left behind. Okay, now I can record the down depth. Now I'm bringing it up, and when I first see those again, I will mark it, and we will record using the tags. The meters are marked with a red, and then these orange lines are 1/10 of a meter. Collect a water sample from this same depth. This is the sample, the collection bottle, and we attach it to the Secchi dish using elastics. Now the goal of this is to collect water at our water sample at the deep depth. So we're lowering it down.
And you get a continuous sample from the surface down to the depth.
Exactly. And it's going down to the depth between the two markers that we've marked before. And we'll wait to make sure it's filled. Carefully bring it up. Now, before we can use the water, we need to take the funnel and the filter, assemble them. We're going to rinse the filters. I have not got that tightly across. Rinse it once, rinse it twice,
This is my deep water that the bottle is 0. We do not rinse the bottle. We rinse the filter. And we're filling to the shoulder. Be very careful that you're not getting your fingers inside the cap or inside the bottle. Now, this is our first go of the season, so we are collecting duplicates. So I have a second bottle with my 0 label. You don't need to rinse the filter again because it's the same site. Now they go into the cooler to stay cool until they are delivered, able to deliver them to my team leader.
So I've just arrived at my nearshore site, and I'm going to rinse my sample bottle before I take the real sample. Okay, I've rinsed it twice. Now I'm going to take the real sample, and I'm going to go down and across and up. Okay, I've now got my nearshore sample. Use this sample water to rinse my filter. Now I'm going to pour this into
You've rinsed it twice. And again, we check the label. This is the right bottle for the site.
and it's filled up to the shoulder.
And add it to the cooler.
The bacteria samples have been collected and must be plated within 24 hours. And you must ensure that the incubator has reached 37 Celsius. Label the collie plate with the same location number that is on the jar lid using the wax pencil. All these instructions are very clearly laid out in the binder. You will now open the jar and then remove the lid from the collie plate. Being very careful not to touch any interior surfaces. Pour the water row by row into the collie plate, filling all the little wells that are there. And you have to check it to make sure. If you hold it on an angle, you'll be able to see if you missed some of those wells, which I have missed a couple right here. Okay, so these wells look all to be full. No, there's another one here. So once the wells are full, You just tap it, little tap, holding it on an angle, and then taking a paper towel or tissue, you just absorb the rest of the water that is just sitting on the edge. Once this is completed and your wells are all full, then you replace the lid, being careful not to touch the inside. And it's ready for the incubator. Your next step is to take the collie plate lid and put blank on it. Follow the same procedure using the distilled water provided to you and fill all of the wells. Once all the wells are full, again, check them.
No.
This is your control. Check the wells. Tap it. And absorb the water that's hanging off the edge with a paper towel or a tissue. Once you're sure that all the wells are full, replace the lid carefully, and it's ready for the incubator as well. You only need one blank, even if you're doing several samples. So the next step is in 24 to 26 hours, we will return to read the bacteria or E. coli levels. You've now incubated your sample for 24 to 26 hours, and so we have the sample, and lift off the lid and check for blue cells. Now there are a number of blue cells here, so it's much easier if you have it on a white sheet of paper and you count those blue cells and you mark the number of blue cells corresponding on that tally sheet. And in the end, it makes it much easier to calculate how many blue cells that you've seen. So say in row 12, there are 1, 2, 3, 4, 5. So you just check those off. This one, this one, this one, and so forth. And then you take your blank and you look at the blank and see if there are any blue cells there. We do not find any blue cells in the blank, which is a good thing. And that means nothing has been contaminated in your session. Now move the collie plate onto a dark sheet of paper, and you have your UV light, and now you're going to be looking at the fluorescent cells. So you have a number of fluorescent cells here. As you can see, don't mistake the little blue things for the fluorescing ones. Take the number, you can chart that number on the tally sheet again. And if you find you have more than 18, it's a good idea to notify the MLA office so they can follow up with whatever might be necessary. After you finish this, you transfer all of the information from the tally sheet onto the data sheet in the blue column and in the fluorescent blue column. Both of those sheets should be returned to the MLA office.
So now we're done with our video, and we'll try and go through some of the questions that have been posted. We have a question asking for the dates for testing again. And so the testing weekends, this coming weekend, May 20th to 23rd. Then in June, 24th to 27th. Then in July, the 22nd to 25th. Then August 19th to 22nd. These dates will be in your binder. So, and it will also, we're going to have this slideshow and video live on the website by tomorrow. We had a question about a slide being just shown briefly, not sufficient time to see it. I'm not sure which slide that would have been. If it was the one having to do with water levels, the water levels are now normal for the summer. There's a question says, when counting E. coli, some cells show very faintly. Should those be counted? Yes, they should. What's really important here is that when we're getting E. coli and fluorescent, they're both blue and fluorescent, equaling 18 or more. We're going to generally ask you to do a retest to see if the condition persists. We're doing that to see if we've got an E. coli count against the part. Please make sure that your incubation time falls within the 24 to 26 hour period. And those tests need to be started within a day of receiving the samples at the team leader's site. I have a question here that says, some of us don't have a binder yet. If you haven't picked up your binder, please either arrange with Chelsea to pick it up at the office or at Seedling Day on Saturday this coming weekend. There's a question, do we need to make observations at all the sites on the May 20th to 23rd weekend? Each sampling binder will have instructions for each site as to what you're supposed to do for each weekend. So read your binder, because some sites that are only bacteria sites will not be tested this weekend. We only do bacteria in June, July, and August. We do phosphorus all months, but we may not do phosphorus at every place every month if it's a site that has had low readings consistently. Also, some sites that have higher readings may request you to do a duplicate sample, but not all sites will have a duplicate sample, so read your instructions carefully to see if that's required. There's a question that says, I can never see much difference in Secchi up and down, especially with waves. How do you estimate those values to a difference of only a few inches? Well, that's good. it's good that you're getting a value that's fairly similar up and down. Particularly because you were reading on the shadow side of the boat. You're not going to read with your sunglasses on, please. And it's good to get Secchi up and Secchi down, agreeing within a few inches. And then you use that Secchi average reading to take your deep water sample. I'm reminded that Seedling Day this year is Saturday at the Port Carling Legion. That's across the street from the community center. It's not going to be at the community center this year. So instead of going left when you go down that street, turn right.
Chris, there's also a question about rinsing the collection bottle the first time you use it. And yes, you rinse the collection bottle at every site.
Okay, so for just to amplify, Jan, we're getting phosphorus samples with the collection bottle, and so we rinse that bottle at every phosphorus testing site. When we are taking bacteria samples, we are not rinsing the bottle. And we're not using the collection bottle. We're using the sample bottles, the glass sample bottles for that. So the bacteria sample bottles are sterilized and you just use them once. Take the sample, put the jar lid on and bring them back. Jan, are you seeing any other questions?
No, I'm looking down and I'm not. Keeps.
Are there any more questions? If you didn't get your question in, feel free to call the office and ask for an answer. And if Chelsea can't answer, she'll refer them to one of us. So thank you very much for your questions and for your attention. Chelsea, if you could move on to the wrap-up slide, please. And if that could be made clear. So to summarize, try and plan your sampling to avoid, if possible, rain. That may not be possible, but and note the precipitation prior to your trip so that our notes are complete. Please note the waves so that your notes are complete. Please note anything else that might affect your readings, construction, erosion, presence of geese or other animals. These is all valuable pieces of information. And believe me, the consultant does look through those to help understand what they saw. Remember to time your deep water sampling to fill the bottle by the time it reaches to the Secchi depth. As we said, the training video will be available on the website for future reviewing. Something we didn't mention, but it's important. We're now trying to keep track of algae blooms. Anytime you're out on the lake, if you happen to see one, it's important that you report it. And the reporting actually has to go not to us, but the Ministry of Environment, Conservation and Parks, who will send out an expert to confirm the kind of algae, because we're really only interested in cyanobacteria or blue-green algae. They can confirm that, and if they do confirm it, then it goes into reporting with the Department of Health. If you have a new site, please take a GPS reading for that site, and please take photos for us. So thank you very much for coming today. Thank you for volunteering. Enjoy your summer. And for those who have time at the end of the summer, you are all invited to a volunteer appreciation barbecue. No, not barbecue, but an event on the Sunday of the last weekend, which ought to be August 21st. Thank you again. And we look forward to seeing you when you pick up your sample kits. And please feel free to call if you have further questions. Take care and have a good rest of the afternoon.